In Tiktaalik rossiei, which is the Rhipidistian transitional fish species and the later evolving species of tetrapods, the extrascapular opercular bones and a bunch of other dermal bones were lost in the skull. One consequence of this is the ability to move the head independent of the trunk. A flexible neck was therefore born. You see that the skull of Tiktaalik is not connected joint anymore with the pectoral girdle. Further changes in the joints between the skull and the spine, which is the craniovertebral junction, resulted in further enhanced head mobility. This is a list of the major modifications in the tetrapod skull. We'll take a look at some of these in the next slides. The previous slide already showed how the loss of dermal bones linking the shoulder girdle and the skull has resulted in a movable head. This was seen as early as in Tiktaalik. The appearance of more mobile joints, synarthrotic joints in the craniovertebral junction, like a condyloid or ball and socket type of joints in the atlanto-occipital joint. And also example is a pivot joint between the atlas and the axis. So that's the atlantoaxial joint, which allows you to turn your head. So this type of synarthrotic joints conferred a wider range of motion to the tetrapod head. Loss of skull bones was also a general trend leading to modern species, with the highest number retained in some reptiles. Birds and mammals also experienced an enlargement of the cranial cavity, reflecting their relatively large brains. We'll also take a look at the existence of mobile joints in the skull, called cranial kinesis, and their functional advantages. Some tetrapods also independently evolved a secondary palate that separates the nasal and oral cavities. So we'll take a look at their functional advantages as well. The last two, the jaw suspensions and middle ear bones, are modifications of the splanchnocrinium, so we'll reserve them for last. So these are some of the backsides of tetrapod skulls, showing the difference in the occipital condyles. The occipital condyles are projections of the occipital bone, like this, for the articulation of the first cervical vertebra. In amphibians and mammals, there are two occipital condyles on either side of the foramen magnum. The mammalian condyles fit with the superior articular facet of the atlas, forming a condyloid joint. In reptilia, including the avian descendants, there is only one occipital condyle. So amphibian skulls have the fewest number of bones among living tetrapods. This probably reflects a derived condition, since early tetrapods are like fishes in terms of the number of skull bones. The bones shaded in green or blue-green here in the diagram are primitive dermal skull bones lost in the modern amphibian skull. Note the loss of many bones in the orbital series as well as the temporal series. As a result of this massive reduction in dermal bones, there is a huge gaping orbit in the salamander skull, for example. And you can clearly see the underlying chondrocranium, like the otic bones there, colored blue, and the palatal. Some of the palatal bones are also seen, the vomer and the pterygoid, when viewed from the top. These are portions of the skull that are normally invisible, covered by the dermal bones. Amniote skulls can be classified based on the presence and the number of temporal fenestrae, also called as temporal fossa. Anapsid skulls lack temporal fenestrae behind the orbit. This is believed to be the primitive condition exhibited by early amniotes, and some believe was retained by turtles. The two big surviving lineages of amniotes are the diapsids and the synapsids. Diapsids have two temporal fenestrae behind each orbit. Synapsids, of which mammals are the only descendants, have only one temporal fenestra behind the orbit. What is the importance of a temporal fenestra? With the growing importance of biting and chewing as a mode of feeding in amniotes, muscles that close the jaws, called adductor muscles, expanded and differentiated. It's hypothesized that the temporal fenestrae provide more room for these adductor muscles to expand and contract during its action, and the rims of the fenestra also provide more leverage for the muscles. The temporalis muscle, for example, is the one lodged in the temporal fenestra of mammals. You can palpate this on your temples. And it is firmly anchored to the upper rim of the temporal fenestra. The other powerful jaw muscle is the masseter, which is anchored to the lower rim of the fenestra, called the zygomatic arch, basically your cheekbone. Such powerful adductor muscles, along with their skull attachments, make it possible for the complex chewing movements that mammals can do. Living anapsids are turtles and tortoises. Some think that their anapsid condition is primitive, but based on their phylogenetic position close to diapsids and the discovery of some fossil turtles that bear temporal fenestrae, it is more likely that the anapsid condition of turtles has been secondarily derived from a diapsid skull. If so, it is reasonable to think that the loss of their temporal fenestrae probably conferred some advantage to the ancestors of turtles that outweighed the advantage of having temporal fenestrae. This is something that's beyond the scope of our lecture. Among those that display a fully diapsid skull are the tuataras and crocodilians. The two temporal fenestrae are located behind the orbit. The upper one is called the supratemporal fenestra, or fossa. The lower one is the infratemporal fenestra / fossa. The bar separating the two fenestra is called the supratemporal bar / arcade / arch. So there are plenty of synonyms. It's formed by the squamosal and postorbital bones. The lower margin of the infratemporal fenestra is called the infratemporal bar / arcade / arch. It is formed by the jugal and quadratojugal bones. The postorbital bar separates the fenestrae from the orbit. Lizards and birds display a modified version of the diapsid condition. In lizards, the infratemporal bar was lost, and so you see a deep notch in the temporal region. The supratemporal fenestra, remains intact, so it's still present. In birds, the supratemporal bar disappears, and so the two fenestrae fused into one. The loss of the postorbital bar also means that the temporal fenestrae became confluent with the orbit, like this. Mammals display a synapsid skull. There is one temporal fenestra behind the orbit, and it may be confluent with the orbit. The infratemporal bar is also called as the zygomatic arch, the cheekbone, which is very prominent in mammals. Some derived features of the avian skull are the following: an enlarged brain case, bulging at the backside. Sutures between the bones also tend to disappear as the bird matures, so the surface looks smooth. The dermal bones tend to be fewer than their reptile relatives, and just like the other parts of its skeleton, it is light and hollow. The jaws have been modified into beaks covered by horny sheath and have no teeth in modern species. The presence of mobile intracranial joints is called cranial kinesis. Many vertebrates actually have kinetic skulls, with the exception of modern amphibians, turtles, crocodiles, and mammals. Kinetic skulls are associated with certain modes of feeding. In general, animals that rely much on chewing or mastication tend to have rigid, akinetic skulls. Cranial kinesis is epitomized by snakes, which, rather than chewing their food in portions, tend to swallow an entire prey, sometimes larger than its head. It is actually a myth that snakes can perform this amazing feat by disarticulating their skull bones. Instead, they simply have highly kinetic skulls. Some of the kinetic properties of snake skulls are listed here. The presence of intracranial joints allows certain bones of the upper jaw to be moved relative to the brain case. An example of this is the rotation of the fang-bearing maxilla, which can rotate, to flare its fangs and inject venom. The quadrate bone there, which suspends the lower jaw, can also rotate relative to the brain case, allowing a wider gape. And this kind of quadrate bone mobility is called streptostyle. The mandibular symphysis is not made of a tough fibrocartilage like in ours. Instead, it is made of soft, elastic tissues, and this allows the left and right portions of the jaw to move independently. This allows the jaws further to kind of walk on the prey as it push the food deep into the pharyngeal cavity. So in the next slide, I'll show you a video to help you visualize this cranial kinesis in snake.
The record breakers for swallowing large prey are almost always pythons, so I'm going to use a python skull for reference in this video. Now I'm going to start off by saying right away that snakes do not unhinge or dislocate their jaws, because that would look like this, and that just doesn't happen. But to really understand what makes a snake skull so special, let's have a look at the skull of something that isn't a snake, like this dog. Dogs, like most mammals, have a solidly built skull, with all the bones fused together, and really only one set of moving parts, the jaw joints. Settle down, Rex. Snake skulls, on the other hand, are more lightly and loosely built, with many moving parts. There are the jaw joints, of course, which are positioned really far back to make the mouth larger, and which are at the end of a pair of long, movable skull bones. The jaw bones themselves are flexible and can bend slightly outwards in the middle, and they're not fused at the chin either, so they can be spread apart. But it doesn't stop there, for the upper jaws can also move independently of the rest of the skull. All this flexibility allows the snake to open its mouth incredibly widely to fit over large prey items, but it also allows the snake to do something else. In order to swallow large things, being able to open the jaws wide enough is only half the battle, for the snake still has to get the food down its throat without the use of arms or hands. And to do that, it moves its jaws independently, grabbing and holding the prey with its backward-pointing teeth and pretty much walking it into its throat one grab at a time.